Senior Advisor, and Dr. Bashnet Singh, Scientist Team. Academic perspective will be shared by Dr. Raghavan Vardarajan from IISc Bangalore, and the industry perspective will be shared by Dr. Mona Lisa Chatterjee, Psyche Bio Private Limited. I now invite Dr. Alka Sharma, Senior Advisor DBT, to set the context for today's session. Over to you, ma'am.
Thank you, Vaishali. Good afternoon. On behalf of Department of Biotechnology and Biorec, I welcome both the experts here from academic setup and also from industry, my colleagues from Biorec and DBT, all the participants who all have joined virtually to this 14th webinar on biomanufacturing of mRNA therapeutics under the webinar series on the Biofoundry and Biomanufacturing Initiative of the Department of Biotechnology under Bio-E3 policy approved by the Union Cabinet under the chairmanship of Honorable Prime Minister. Released by the Honorable Minister of State, Independent Charge, Ministry of Science and Technology, Dr. Jitendra Singh Ji, this policy aims to build a resilient biomanufacturing ecosystem in the country for developing bio-based products. The Bio-E3 policy is aligned with the Honorable Prime Minister's clarion call for lifestyle for the environment, that is life, and the Union budget emphasis on green growth. The interim Union budget for 2024-25 had also announced Biofoundry and Biomanufacturing as a new scheme. As you all know, the Department of Biotechnology along with Biorec have played a pivotal role in nurturing and robust biotechnology ecosystem across the country. Through these efforts, India is now poised to join a new global wave of industrial revolution, focusing on more sustainable manufacturing practices driven by recent advances in biotechnology. As you are aware, global challenges such as climate change, unsustainable patterns of material consumption, and waste generation require concerted, sustainable interventions. High-performance biomanufacturing has the potential to provide solutions to these challenges by fundamentally transforming the global economy to the regenerative principles. Building on this transformative potential, the emerging biorevolution could have a significant impact on bioeconomies and our lives by paving the way for a wave of sustainable innovation across sectors. Through extensive national consultation and sectoral experts meeting by Department of Biotechnology along with Biorec, 6 thematic sectors have been identified for implementation under Biomanufacturing Initiative. They are bio-based chemicals, enzymes, APIs and biopolymers, smart proteins and functional food. The third one is precision biotherapeutics. This includes cell and gene therapy, mRNA therapeutics, on which we are going to have discussion today, and monoclonal antibodies. Fourth sector is climate resilient agriculture. Fifth one is carbon capture and utilization. And we also have added the futuristic marine and space research and exploratory sector under Bio-E3 policy. I would like to share with you that the calls for inviting proposals in many of these thematic sectors for fostering high-performance biomanufacturing under Bio-E3 policy have been announced. Regulatory conclave are also being organized to develop a framework for coordinated regulatory reforms for each of the thematic sector and subsector. In this regard, a regulatory conclave on smart proteins has been organized in consultation with all stakeholders. Further, a stakeholder consultation on emerging regulatory pathways for cell and gene therapy products have also been organized. DBT Biorec Mulankur Bioenabler is an essential component under Bio-3 policy. Under this, a network of cutting-edge bioartificial intelligence hubs, biofoundries and biomanufacturing hubs are being set up across the country to augment discovery and application-oriented integrated network research, bridging the gaps and scaling up between the lab to market, and for pilot and pre-commercial scale manufacturing across the thematic sectors and subsectors to accelerate India's progress towards becoming the global biomanufacturing leaders. The proposals for setting up bioenablers that received under the first call are under implementation, whereas the call is also open to invite proposals for setting up a biofoundry and biomanufacturing hubs and also bio-AI hubs. For bio-AI hubs, the aim is to address complex biological research challenges using data-driven, cutting-edge, multidisciplinary, programmatic research initiatives. I would like to share with all of you that the India's first biomanufacturing institute, that is the National Agri-Food and Biomanufacturing Institute in Mohali, has been dedicated to the nation by the Honorable Minister last year, October. In order to promote research in space biotechnology and biomanufacturing, which is an important vertical of Bio3 policy, an MOU has also been signed between DBT and ISRO. And a joint working group has also been constituted to identify joint research proposals. Coming to center-state partnership, I would like to share with all of you that this is an important component of this policy. So for this, a center-state partnership conclave was organized in February this year under the chairmanship of Honorable Minister. The discussions were held with the representatives from 24 states and union territory, science and technology departments in the state, councils, industry experts, senior officials from DBT and BIREC. During the conclave, state governments were asked to set up Bio3 cell to facilitate biomanufacturing initiative aligned with the local priorities and resources while remaining connected with the national objectives. As a follow-up of this conclave, an MOU has been signed between DBT and Government of Assam to formalize a strategic collaboration in which DBT will extend its guidance and facilitate partnerships, while the Government of Assam will spearhead initiatives by establishing a state Bio3 cell to implement Assam Bio3 Action Plan. With this, Assam has become the first state in the country to establish a state Bio3 cell. Outreach activity for Bio3 policy are also being carried out extensively through press meet, social media platforms, editorials to highlight its significance for the country. You may like to visit the biomanufacturing website for the detailed activities which are being undertaken under this biofoundry and biomanufacturing initiatives. To further disseminate the information regarding various thematic verticals under Bio3 policy, DBT jointly with BIREC has launched a webinar series on biofoundry and biomanufacturing initiative, and this series features discussions by the experts on thematic sectors and subsectors, DBT and BIREC officials, emphasizing their potential impact on India's bioeconomy. It provides a valuable platform for researchers, startups, SMEs, and industry representatives. Today's webinar is the 14th in the series, focused on biomanufacturing of mRNA therapeutics. Several Indian biotech companies and research institutions are actively involved in developing mRNA-based vaccines and therapeutics. Increasing burden of cancer, genetic disorders, and infectious diseases is driving the demand for these precision therapeutics. However, in today's webinar, some of the challenges such as product delivery, stability, availability of raw materials, and its local supply chains and appropriate regulatory framework will also be discussed and addressed by the experts, which we have today with us. So with this, I would like to close my remarks, and I will warmly welcome each one of you to the webinar on biomanufacturing or mRNA therapeutics. Looking forward to a fruitful discussion today. Thank you. Thank you very much.
Thank you, ma'am, for providing an excellent overview of the vision and the objectives of this initiative. Our next speaker is Dr. Vashni Singh, Scientist D at DBT, who will discuss briefly about the biomanufacturing from mRNA therapeutics from the DBT's perspective. Over to you, Dr. Vashni.
Thank you, ma'am. And good afternoon, everyone. I hope my slides are visible.
Yes.
So I will briefly give a primer to this vertical, mRNA therapeutics, before our esteemed experts take over. So mRNA therapeutics, as we all know, is a very powerful and versatile tool to combat diseases as wide-ranging as cancers, genetic disorders, and infectious diseases. And the global market for mRNA therapeutics is currently dominated by North America, with over 40% share in 2023. And this was driven largely by the sales of approved mRNA vaccines for COVID-19. Several Indian companies like Biological E, Bharat Biotech, and Genova have also ventured into this space. And Genova had also launched mRNA COVID vaccines in the market. While this field shows immense promise, there exist certain challenges, as already mentioned by Alka ma'am, such as delivery systems, product stability, and regulatory approval. And if these challenges could be overcome, then mRNA therapeutics will be a cornerstone of precision medicine as we move into the future. So this slide just lists out the significant path we have covered over the past few decades, with the discovery of mRNA in the year 1961, and several important discoveries and advancements in this field have happened since. The first mRNA-based company was founded way back in 1997, followed by several other clinical trials with these mRNA products. And during the COVID-19, we saw 2 mRNA vaccines being introduced into the market for the very first time. This also resulted in the Nobel Prize for Medicine in the year 2023 to Dr. Katalin Kariko and Dr. Drew Wiesman for their discoveries concerning nucleoside-based modifications that enabled the development of effective mRNA vaccines against COVID-19. There are several steps in the mRNA drug production pipeline, which begins with the first step, which involves designing the mRNA, followed by synthesis and purification. The next crucial step is mRNA entrapment, and this has several options available in the form of lipid nanoparticles, cationic liposomes, and dendrimers, followed by pharmacodynamics and safety and evaluation in vitro and in vivo in animal models, followed by manufacturing and clinical trials of the final end product. So the department had carried out a SWOT analysis of this sector, and we can see that there are several strengths within the country, such as a vast network of public and private R&D institutes, availability of an indigenous mRNA platform. We have significant expertise in IT and nanobiotechnology within the country. And we also have a vibrant startup ecosystem to top this. There are several opportunities as the burden of cancer, genetic disorders, and infectious diseases is growing within the country and worldwide. And with the introduction of the Bio-E3 policy, there has been a significant policy push for biomanufacturing of indigenous products. And there are avenues for advanced technology development in fields such as microfluidics, AIs, etc. However, we have certain weaknesses which we want to address as we move forward, such as the lack of local supply chains for critical raw materials and absence of clear regulatory pathways to take these products forward. And there are certain threats also, which is mostly due to the import dependence for the raw materials. So as was already mentioned that the government of India, led by the Department of Biotechnology, came out with the Bio-E3 policy last year. And precision biotherapeutics was identified as an important vertical. And mRNA therapeutics is an important part of this. So taking this forward, DBT BioRec have jointly announced the first call for proposals in the area of precision biotherapeutics, mRNA therapeutics, under 2 categories, which is discovery and application-oriented integrated network research and bridging the gaps for scale-up. And the last date for submission of proposals has been extended till 31st of May. So this call is currently active for any investigators who want to submit proposals in this particular thematic. Thank you.
Thank you, Dr. Varshney. It is now my pleasure to welcome Dr. Vardharajan, ISC Bangalore, to present the academic perspective. Dr. Vardharajan obtained his PhD at Stanford University, and following a postdoctoral stint at the Yale University, he returned to Bangalore in 1992 to start his academic career at ISC Bangalore, where he is currently professor at Molecular Biophysics Unit. His laboratory works on protein stability and folding and its application to design immunogens to protect against major viral threats, including HIV-1, influenza, SARS-CoV, and Sebrico In 2017, he co-founded the startup Manvax to advance his vaccine work to clinical development and commercialization. Dr. Vardharajan has been conferred with several awards, notably B.M. Birla Science Award, Swarn Jayanti Fellowship, Santi Swarup Bhatnagar Prize, J.C. Bose National Fellowship of BST, G.N. Ramachandran Gold Medal for Excellence in Biological Sciences and Technology, and the TWAS Award in Biology. He is an elected fellow of the Indian National Science Academy and the Indian Academy of Sciences. Over to you, sir.
Thank you. I'll just share my slides. Thanks for the kind introduction. Now, what I'll be talking about here really is not really biomanufacturing, but our experiences in just setting up lab-scale mRNA LNP synthesis and testing over the past couple of years with funding from Ignite Life Sciences. And this is really directed at people in academic labs or other startups who want to start in this kind of work. Now, there are a lot of important advantages of the mRNA vaccine modality, and these include relatively rapid manufacture and the fact that one mRNA molecule is much like another. So process development is much easier compared to a protein vaccine. And also one can, in the vaccine context, you can stimulate both cellular and humoral immunity. And so these are the advantages. Of course, there are some disadvantages. So relatively high reactogenicity, which makes repeated administration challenging, temperature instability. And in the context of vaccines, if the immunization technique is not proper, you can get systemic expression of the antigen. And it's difficult to characterize the expressed protein product because it's produced in very small quantities. Now, the steps involved here are the mRNA synthesis, the encapsulation, and in the case of vaccines, immunization. In the case of therapeutics, there would be other downstream modalities. So as far as the mRNA goes, for those of you who are not completely familiar, the very important elements are the 5' cap structure, the 5' untranslated region, the open reading frame, and the 3' untranslated region, and the poly A tails. And so the cap structure is important for stability, and it needs to be added either during or post in vitro synthesis. The optimal choice of codons to maximize expression also has to be worked out. And this is actually quite a challenging area because for a given protein sequence, there are very, very large number of potential mRNA coding sequences. And finding out which might be the best ones amongst these is a challenging job. And there are no really clear rules which at the present time allow you to do this. And yes, so these are the things that need to be optimized, the UTRs, and there are now a bunch of UTRs which are available, and also certain modifications one can make in these. In the case of secreted proteins, there's optimization of the signal sequence. And as I said, one of the most challenging things is how to optimize the codon usage and to both maximize the translation and to allow the sequence to adopt a stable structure for increased stability both during storage as well as inside the cell. So during the process of in vitro transcription, one can either start out with a linearized template, which can be achieved either through single or double digestion from a plasmid or through PCR. For manufacturing, of course, you would like something which is involves probably does not involve PCR. Then there's the step of in vitro transcription, which is typically done with T7 RNA polymerase, either the natural one or a modified one, and using nucleotide triphosphates, which can either be the natural or in many cases now preferably the UTP is modified and T7 polymerase and buffer. And then, as I mentioned, there's a cap structure. And so the cap structure can either be added enzymatically, post-transcriptionally, or co-transcriptionally. And so there are good cap analogs which are available for this, but there's IP associated with it. And on an industrial scale, there may be quite high costs associated. So our experience, we made our own T7 RNA polymerase. And what we found is that having additional chromatographic steps other than just the histag purification leads to a substantial improvement in the quality of the mRNA. So there's much less of the shorter products and increase in the expression in mammalian cells also. So, and again, this yield is only indicative of a lab scale process. It can be much, much higher in a fermenter. So the next step is to make the lipid nanoparticles. And I will be talking here about the conventional lipid nanoparticles. There are many other modalities which have been mentioned already by Dr. Varshnaya, but for the most widely used are the standard linear mRNA with lipid nanoparticles. And that's what I'll be talking about because these have the most safety data associated with them. So typically one makes an ethanolic solution with the lipids, and then there's an aqueous buffer containing the mRNA, and then these need to be mixed. And they can be mixed by a variety of different methods, either manually through a pipette pen or through ethanol drop precipitation or microfluidic chips or through a mixing device. And we have explored all of these. The most important component of the lipid is the ionizable lipid. It's also the most expensive. We were fortunate to have a collaborator, Professor Mridmoy Day, who synthesized these lipids for us, the ionizable lipids. All the others are available commercially at not too large of a cost. So now if one looks at the different methods of encapsulation, the three methods I talked about, we found the best results with the microfluidic chips. I'll come to the nanoscaler in a moment. And you can see that this was with the luciferase mRNA. And so the amount of reporter gene expression we got was highest with the mRNA made by the microfluidic method. And so after some initial testing, for a while, we were looking at the microfluidic chips. Now, the problem is, so we were also trying to fabricate our own chips, and we had a very good fabrication facility at IISc, at the Center for Nanoscience. The problem was that these fabricated chips could not withstand very high flow rates, and there was often leakage and clogging, and we would get very inconsistent sizes and the PDIs, the polydispersity index of the mRNA LNPs. Next, we tried glass chips, which could withstand high flow rates and are reusable, but we found at least the ones we tried had severe clogging and they were prone to breakage. And again, we got inconsistent sizes because of that. Then finally, we looked at polymer chips, and here we got the best results. And they could be used, they could be reused. They didn't clog up that easily, and we were able to get relatively reproducible sizes of the mRNA LNPs. So the setup here is quite simple. There are two microfluidic pumps, and one contains the mRNA in aqueous solution, the other lipids in ethanol, and these are flown through this commercially available chip, and then you collect the output. We also subsequently, with some funding that we were able to get from the Gates Foundation, we got this mixing device from NAWA, and we looked at both of them. With the microfluidic, so we looked at a bunch of different chips, and we got, again, the best results with the COP chips. And but the nanoscaler gave us much more reproducible results and a much smaller size of nanoparticles. So typically you want to have nanoparticles which are of the order of 100 nanometers or less in diameter. And encapsulation in all cases was quite good. So this just summarizes. So we found that the mRNA LNPs made by the IgM mixer were the smallest and most homogeneous. And although the initial investment in getting the system is a little higher, but then there are no running costs, or very low running costs, because there are no chips that you have to replace, and unlike some of the other commercial systems. So we were quite happy with this. So we did, just to show, we did an immunogenicity study with, we wanted to compare the kind of immune responses we were getting with mRNA. This is the ALC0315 ionizable lipid, which was used by Pfizer and BioNTech, and with a protein formulation that we had developed in the context of COVID, a thermostable protein adjuvanted formulation. And so we gave mice 2 immunizations, and these are the ELISA titers. They were somewhat higher for the protein compared to the ALC0315 mRNA, but both were pretty good. And yes, so that summarizes what I just said. Then we looked at the pseudoviral neutralization titers and did a mouse challenge. So again, there's a prime boost, and then we did a viral challenge with mouse-adapted virus. So the neutralization titers, like the ELISA titers, were roughly 1 log higher with the adjuvanted protein formulation. Both groups were protected from challenge in contrast to the unimmunized mice, but the weight loss was a little bit less for the protein formulation. So again, the protein formulation in our hands worked a little bit better. And of course, it was thermostable. So just to summarize this very brief talk, we were able to make mRNA with the in our lab-prepared T7 RNA polymerase and encapsulate it. And we found in our hands the co-transcriptional capping was more efficient than the enzymatic capping. And one of the big problems we encountered was very poor quality of enzymes supplied by local suppliers, even though these were imported enzymes. So sometimes they would work and sometimes not. The modified mRNAs, which modified nucleotides, showed better in vitro expression. For a low-cost formulation device, microfluidic pumps and the COP chips work the best for us. But finally, I think now we're largely using the NAWA system, and we were able to get a protective immune response. So just to thank all the people from my lab and my collaborators at IISc, from Nirmoy and Siddharth's labs, new assays from IMTech, the adjuvant from BFI, and some funding from the Gates Foundation, and Minvax for a lot of help with all the animal studies. So thank you. I'll stop here.
Thank you, Dr. Vaid.
Should I stop sharing the screen?
I would now like to invite Dr. Mona Lisa Chatterjee, Psyche Bio Private Limited, to present the industry perspective. Dr. Chatterjee is the co-founder and the director of Psyche Bio Private Limited. She received integrated MSc and PhD from IISc and carried out postdoctoral research at the Yale University, USA. She has led diverse drug development programs in organizations, namely AstraZeneca, Biocon, Kinsam, and Bill and Gates Foundation. Few of these drugs are approved today. In 2022, she was recognized among 75 women by the Government of India for excellence in the field of science and technology. Dr. Chatterjee has 20+ years of experience, both in small molecules and biologics discovery and development. With this, I invite Dr. Chatterjee.
Thank you so much, and thank you for the kind introduction. And it's a pleasure to be here and listen to all the experts. I'll try to show my screen. Let me know if it's visible.
Yes, it is visible.
Perfect. Thank you. So I think I was given the mandate to talk about how we visualize India being a global hub for precision medicines. So we've taken where we are today from the industry point of view and where would like to be together as a scientific community grow so that we can make high quality disruptive precision medicines from India. So I'll take a little bit step back to quickly say what is a biologic. We have heard very nice presentations. Why is biologics more complex? Because unlike small molecules, they are derived from biological systems. Hence, they are inherently large, diverse, have modifications. They can be composed of sugars, proteins, nucleic acids, or combinations. So just by the name, they are complex. And they come in many different flavors: vaccines, hormones, enzymes, proteins, etc., etc. If you look at biologic therapeutics, they really treat, and Dr. Sharma, Dr. Varshney, Dr. Varadarajan all mentioned, they are becoming more and more important in the physicians' arms and armamentarium, that they treat cancer, diabetes, rheumatoid arthritis, and many other critical chronic diseases, as well as infectious diseases. The reason why they're gaining popularity, they are highly targeted and very efficacious. Big challenge with biologics is they're very expensive. So a large fraction of global community and geographies, it's really difficult to access. Though we have the life-saving drugs, it's difficult to access them when they are needed for the patients. If you look at it, if you look at the biologics market, it is the United States and North America which comes out as the major market for, and rest of the world really doesn't have any penetration of biologics in the rest of the world. So we started with the fact that how can we have an impact and how can we change or do our small bit of contribution to change that. So looking at why are biologics expensive, there's high cost of discovery, they're complex to make, they need high capex or the large facilities to make them, very high manufacturing costs. Everything, many of them has been mentioned. They have long process time cycles to make it, and storage and distribution is a challenge, something Dr. Varadarajan, Dr. Varshney alluded to. They're not very labile. So either you need cold chain or you need low temperature. So they're not very stable molecules. What is the opportunity? Now, Dr. Varadarajan mentioned a lot about linear RNA. Now, linear RNA has really shown its tremendous potential of scale with COVID vaccines. However, it is a modality which is intrinsically more labile. And also, from our point of view, this is something Dr. Varshney mentioned, there is an extreme competitive space, and the IP landscape is quite complex. So as a young group starting in India and wanting to make a difference, we decided we will work on the second generation or next generation modality of RNA, which is a circular RNA, which is nothing but a ring structure. You have removed the ends and the molecule is intrinsically more stable. Also, the advantage is that it is a right time for innovations in this space, such that we can have our own IP in-house technologies that can be developed. So the circular RNA, as I mentioned, is more stable, does not require cap, right now does not need any modified NTPs. However, there are multiple innovations that are needed to make it really a good platform. So what does RNA do? RNA is put in the body. Instead of putting the therapeutic protein, the body looks at the RNA and makes the protein. It looks quite simple. But your RNA should be delivered at the right place and should be in sufficient amounts so that you get the therapeutic at the right dosing, as well as you should be allowed to do repeat dosing. The advantage of RNA is all the decorations of the protein are human-like, making it less immunogenic. RNA intrinsically is a low capex, low footprint manufacturing platform. And it is truly a platform because you can develop multi-product facilities in this space. with a very fast cycle times. So you see, from the manufacturing side, it is really attractive to convert all biologics in a really blue sky way to RNA and really make these life-saving drugs accessible to patients globally. So that is where we thought we should aim for. Circular RNA can be made into various different use cases. You can develop stable multi-subunit vaccines. You can ideally, if you can build a platform, you can deliver antibodies. You can deliver enzymes or hormones for, say, rare diseases. There have been applications in gene editing. And of course, it can really simplify CAR T therapy, CAR T manufacturing, as well as in vivo CAR T. However, while on one hand, there is immense potential, on the other hand, there is also aspects that needs to be built into the technology platform. You absolutely need to build a commercially viable process, which is out of the research space, that is scalable, and that can have the right analytics associated with it. And that should always be done with respect to quality as well as cost in mind. And our continuous endeavor is how we retain the quality and bring down the cost. You have to make stable RNA, also stable formulated RNA. And if you want to build a therapeutic out of it, they should not be immunogenic, as Dr. Varadarajan alluded to. And they should have enhanced shelf life and portability. Aims to enhance protein production from a single RNA will have a significant impact on the dose, safety, and cost again. So if you enhance expression, you can actually make it a much better platform. If you can express more protein from one single RNA, again, you will open up biologics like monoclonal antibodies delivery as an RNA. We need improved delivery systems, as I mentioned. Also very important is we need to develop in-country technologies so that we do not end up having high costs due to very high in-licensing fees when we in-license technology platforms. This is what our vision is, and I'm going to, since it's just a 10-minute talk, I'm going to just run through quickly. We started from we want to develop an integrated circular RNA workflow. We started with design. We have our own proprietary AI, ML, and physics-based design platform that is validated today, is used for both therapeutic design as well as immunogen design. And what we have been able to do with that is have novel proteins created that are for various different use cases. Post that, we would like to take the proteins and deliver it as RNA, and we have built our own circular RNA platform, which has novel means of circularization, does not have byproducts, something what Dr. Varadarajan showed. It does not have byproducts. It allows very precise junctions and also recircularization of the linear. With that, we have built analytics to very strongly look at what is linear and circular. A scale-up ready process has been established, and we have also achieved expressing both multi-protein from a single RNA. Where we would like to go, which is what going to really have an impact on biomanufacturing, is backward integrate nucleotides enzymes, because a research level process is very, very well established. Have a semi-continuous process and have engineering solutions for contactless manufacturing. On the LNP side, while there are many gold standard LNPs available, Dr. Varadarajan did mention one, we use very routinely Moderna-like formulations. However, they are, what we would like to go is non-infringing or proprietary stable formulations. And that is an ongoing work that we are doing in-house. We have had the same experience as he shared. We now have our own fabricated LNP device. However, what is very critical for us is at commercial scales, how do we scale it up? What's the right device and what's the right methodology to scale the formulations? Of course, finally, the fill finish would be something we will always outsource. So the programs that are running, we have used many different research level proteins like reporter genes like nanoluciferase. We have used COVID vaccines, both spike and N protein as immunogens. and we have also used other reporter genes. But the two active programs that we are developing in-house and with the development of the platforms are an RSV vaccine candidates, which we are looking at circular RNA-based, which is now in preclinical, where we have both known immunogens as well as novel immunogens designed by our platform. And the second program we are developing is an anti-HER2 antibody being delivered as a circular RNA, just entering animal studies. We have very good data in cell lines. I would like to acknowledge Bill and Melinda Gates Foundation for their support in this programmatic work. So if I just quickly look at taking this technology, we are very established in the lab, and how do we now take it to really a commercial and manufacturing scale? What are the bottlenecks? Actually, there are limited manufacturing capabilities today. And more importantly, it may be easier to set up a plant. There are not enough products near commercialization in near space. So how do we think about commercial manufacturing as our product pipeline is maturing? For therapeutics, higher dose will be required. Capacity has to be higher. We do not have trained manpower as of now. We really have to upskill our manufacturing as well as bioprocess scientists towards RNA manufacturing. As a community, we don't have experience scaling up the technology. We are learning as we are going ahead. We have experience with proteins and peptides and monoclonal antibodies, but not with this modality. What was mentioned really is RM procurement. Now, DNA, linear DNA, PCR DNA, doesn't matter, is a raw material in starting point of this process. There are other critical RMs. It was shown to you, the enzymes are one. Circular RNA has less number of RMs, that's good, but still you have to know how we will have raw materials that are quality raw materials and are also sustainable for a commercial manufacturing. And how do you get the consumables that are really aligned with RNA manufacturing is a big space, which is often overlooked. Okay, I'll just leave with the thought that if you really have to compete and stay with the global development in the RNA technology commercialization, significant capital is required. And it's required on various different areas: research, product development, manufacturing capability, capacity, and training our own people and ourselves towards this new modality. So with that, I would like to say thank you from our team and thank you for this opportunity.
Thank you, Dr. Chatterjee. We will now open the floor for questions from our audience. Please type your questions in the chat box, and our team of moderators from DBT and BIRAC will assist in facilitating the discussion. Let us take the first question.
Question is for Dr. Vadrajan, and it is from Dr. Neha. What technical adaptations are needed to expand mRNA platforms beyond vaccines to treat chronic diseases or genetic diseases?
Right. So I think one of the issues is, with vaccines, the amount of gene product that's required is very small. So, that's a relatively low barrier to overcome. With therapeutics, you need much larger output of gene product, and you also need repeated injections. And so issues like reactogenicity also have to be dealt with. So, and of course, targeting is a much more crucial issue for therapeutics. Vaccines, just intramuscular immunization, and then the expressed gene product is taken to the lymph nodes. So it's much easier. But with therapeutics, these are at least some of the barriers that need to be overcome.
Thank you, sir. So the next question is from Dr. Donald James, and it is for Dr. Alka ma'am. Dr. James is asking, mRNA therapeutics being a novel class of therapeutics have to undergo a thorough and uncertain regulatory pathway. So what steps are DBT and BIREC taking in this direction?
Thanks, Amit. And this is very, very important question because as I mentioned in my remarks also, this is something is a independent vertical, the regulatory framework, appropriate regulatory framework that is needed for each of the verticals. So whatever these sectors and subsectors have been identified, we are going to organize a regulatory conclave and with all stakeholders just to know that what exactly is required to bring the product to the market. And well before that, also all the three categories, basically scaling up, that is bridging the gap, pilot scale and pre-commercial, before it is reaching to the market. So, for example, we have done this exercise in a smart protein sector and jointly with FSSCI, so DBT, BIREC and FSSCI, we organized this conclave. We received really good recommendations and that we have to jointly address those recommendations. So we are taking that ahead with FSSCI. Similarly, mRNA therapeutics, which we are discussing today, this is important to discuss each and every component of this in terms of that bringing the final product to the market. This is very, very important and critical part of this entire chain and this pipeline that has to be completed. Without regulatory framework, appropriate regulatory framework in the country, we are not going to resolve the issue. So this is something that we need to address. The DBT and BIREC jointly will do this with the stakeholders and experts, and that is our plan. And similarly, we are going to have in all the sectors which have been identified under Bio3 policy. Over. Thank you.
Thanks, ma'am. So the next question is for Dr. Chatterjee, and it is from Dr. Richa Kapoor. And she is asking, what are the critical gaps in scaling from preclinical to commercial scale production?
Yeah, thank you so much for asking that. So when you say, commercial scale production really is all about how we can have a seamless process, analytics, and what would be the properties on which you will actually accept your product, final product, as well as both the RNA, which is your drug substance, as well as the formulated drug product, right? So there is globally there is a draft guidance on linear RNA, but there is actually nothing on circular. But we are emerging, trying to understand what are the key properties on which we should assess our RNA and accept it. And I think that is one. Analytics is a very, very important aspect. The second aspect is the raw material. How will you get all the raw material? How will you qualify the raw material that will go into your production? Third is, how can you make your manufacturing yield RNA, which is at high yield and the right, size, quality, no aggregates, etc., etc. So how do you make the process of manufacturing efficient as well as effective? How do you remove the byproducts? So when we do at research scale, we really are trying to get some purified material to do our proof of concept. But when you are taking it to a commercial scale, like for any other, so interestingly, the RNA scale, if you compare to MABS production in a 5,000 liter bioreactor or a 2,000 liter bioreactor, a manufacturing scale of RNA is in 100 or 10 liter. So the scale is amazingly different, but the quality and the science behind it stays the same, right? So you need to look at process efficiency. You need to look at analytics and how you will qualify your product, and you will need to look at the raw material that comes into your product.
Thank you, ma'am. So the next set of questions is with BIREC. These are from the industry participants. Over to you, Dr. Sujit.
Yeah. Thank you. Thank you, Dr. Amit. So the next question is from Dr. Pakshirajan. This is for Dr. Vardarajan. Sir, the question is regarding the formulation and delivery. So they're asking basically the scaling up of the lipid nanoparticle encapsulation, which is quite challenging due to sheer sensitivity and mixing constraints. So how do one take care of that?
So, I'm really not qualified to talk about scale-up issues because we haven't done any. But, using very basically near identical things to what we have used, BioNTech has also used similar kinds of, mixing devices from now, and they have been, Pfizer and BioNTech, and they have been able to produce very large amounts of material. So I'm not sure that, at least for the mRNA LNPs, I think a lot of these scale-up issues have been worked out with not incredibly complicated equipment. Of course, now as regulators become more stringent and more demanding on the quality, as Mona Lisa said, there has to be probably better analytics to characterize many of these things. But per se, I don't think that the scale-up is, it's already been done. I don't see that it's a huge problem. For the mRNA, for other modalities, it's different.
So thank you, sir. Dr. Chatterjee, do you have to add any point on this?
Yeah, so I will qualify. I have my colleagues who are the formulation experts. I must say that there are different modalities being used. People have used scale out, so they're parallel processing of a smaller modules. But what we have seen is, and we are learning in this space, different ways of creating the LNPs have different scalability. And I'm sure there are many in the audience who will have better expertise. And while it's served forward process, I think there are lots to be worked out with respects to scaling up the technology, especially if you're going to go into therapeutic space. Do we know everything today? And my limited knowledge, I'm being very careful here, I don't think so. And I think many different organizations, as we come in conversations, are trying various different methodologies to scale it up. With respect to both creating formulations that meet the specifications, that deliver the RNA to the appropriate place, and are also very stable. Please remember, now the whole game has shifted. You want stable RNA LNPs. You don't want something that needs a -80 at all the time. It will not work in our geographies. So, so there is, I think the space will still evolve is my last. I don't think, I will differ a little bit. I don't think it's fully understood with respect to therapeutics in mind.
Thank you, Dr. Chatterjee. So I think the participant is satisfied with the response. Thank you so much. Next question is for Dr. Alka ma'am. Ma'am, the question is from Dr. Saikia. The question is, mRNA therapeutics production is a quite costly affair. So how will BIRAC and DBT support startups in this direction?
Thank you. So the government of India through DBT has already approved the budget for biomanufacturing under Bio 3 policy. That's a separate proposal that for biomanufacturing initiative where mRNA therapeutics is one of the sectors. So therapeutic, actually that is very true that it is very expensive and that this is the right time to bring the technology to the country. So basically, the proposal, the research which Department of Biotechnology along with BIRAC, which we are looking forward to receive proposal in the 3 categories. The first is discovery and integrated network research. That is something the discovery research is somebody would like to do. So that call is already open. As Vashna said that the last date is 31st May. That will cover 2 categories, that is discovery and integrated network research. The second one is bridging the gap for scaling up. So whatever research that startup has done, the proof of concept or any leads that startup has at the laboratory level can be scaled up. And for that, biofoundries are being set up across the country and startups are free to use those biofoundries, and for which also the calls will be announced to receive the proposal from the startups to use the facilities. That is one. The second is that this policy actually covers up to pre-commercial level. So feasibility studies, validation, testing, everything has to be done at the initial phase. And once it is ready up to pilot and pre-commercial, industry has to come forward for manufacturing. So this is the entire pipeline, right from a discovery, innovative research up to pre-commercial. And after that stage, the industry has to come forward for manufacturing of this. So it's a long pipeline that where everyone who all are the, especially for a startup, the question is for a startup. So there is a 50 lakhs grant for the initial phase for which the call is open. So that is, there is no contribution from a startup or industry. Beyond 50 lakhs, the industry, especially for biomanufacturing hub, I am just talking about 30% contribution has to come from industry jointly with Government of India through DBT and BIREC. So that it will be co-funded projects, and basically the team has to work together to bring the technology to the country. Once the technology will be developed in the country and manufactured in a mass scale, we all are sure the cost will go down. So we all have to work collectively, jointly, to bring the technology in the country. Make in India approach has to be adopted in this case, and this is very, very important. And I think it's the right time to do this, and we all have to work together towards this. Over.
Thank you, ma'am. Thank you so much for your response and clarifying this. Thank you, ma'am. Next, I'd like to request Dr. Dheeraj Kumar from BIRAC for a question, please, sir.
Thank you, Sujit. My question is for Dr. Varadarajan. Sir, can you tell about the practices for evaluating RNAs in the production facility? So what kind of measures and what kind of test you are taking for the measuring RNAs? Sir, you are not audible.
Sorry, could you repeat the question? I couldn't hear it very clearly.
Sir, I just wanted to know about the practices for evaluating RNAs in the production facility.
Oh, I'm not qualified to talk about that. We just do lab scale work, so I couldn't. I can't. Maybe Mona Lisa can comment about that. You're muted, Mona.
I don't know. Is it RNA or RNAs? I'm sorry, I couldn't hear.
It is RNAs.
RNAs. So we right now have very good practices of looking at RNAs. We are not a production plant. I want to clarify. We are at a pilot scale. So you are asking somebody who's not at the commercial scale how the RNAs is monitored. I'm assuming your question is in surfaces, glassware, process, media. Is that the question you are asking as a contaminant?
Yes, ma'am. Actually, means RNAs is very ubiquitous in nature, and it
Absolutely.
can degrade. If a small amount of RNAs can degrade the messenger RNA. So, during the production facility, means what kind of practices means in industry are taking care for measuring that is it the RNAs is present or not?
Yeah, so there are standard practices with R, and I think everybody will say, DPC treated all of them. You have inhibitors throughout. You treat all your glassware, but so that is standard practice, and we don't get RNase contamination with all the practices where you have made sure your glassware is treated, your RNs are checked. So every raw material we buy, doesn't matter, lipid doesn't matter, we actually check it in-house. You have assays for RNAs, you have assay for endotoxin, you have assay. So those are the standard practices. And that has helped us in the pilot scale level to not have contamination from RNAs or wipeout batches. Our analytics, you asked something about analytics. We have our own HPLC methods or chromatographic methods. We are we can very go in and actually precisely tell you how much is the concentration and the percentage purity of circular RNA versus linear RNA, because we primarily focus on circular and how we are enriching it. And for visualization tools, you have gel electrophoresis and other methods to visualize your RNA. Right, so that practice is there. At the commercial scale, if you're asking the question, I'm probably not able to give you the answer today. Maybe as soon as we have a plant, I can give you the answer, but today I don't have that.
Yeah, but I actually think
question.
It'll be less of a problem, at the commercial scale than at the lab. Especially at the academic lab scale, we don't have you can see my background, it's not very sophisticated. And we are able to manage without, occasionally we do get contamination, but, in a proper GMP facility, I don't see it as being a huge problem.
Okay,
Thank
sir.
you. Yeah, thank you, sir. So the last question of the day, I just request Dr. Smita to please facilitate.
So this one is for Dr. Varatharajan again. Mr. Bhatt is asking, so you have already alluded to this during your presentation. He's asking what factors or characteristics influence the non-specific immunogenicity of the mRNA? The immunogenicity.
I wouldn't say it was non-specific. It's just that when we compared it with the adjuvanted protein formulation that we had, the immunogenicity was a little bit lower, but it was quite antigen specific. There was no non-specificity. So it could be either the quality of the RNA or the fact. You see, the mRNA LNPs don't have any external adjuvant. It's the LNPs themselves that have an adjuvanting property, whereas our protein formulation had an adjuvant. So it's a little difficult to compare the two. We've just compared them at the same dose in micrograms, but that's not a very meaningful comparison.
Okay, sir. Thank you for your answer. And with this, I think we'll close the question and answer session, and I will hand it over to Dr. Vaishali Punjabi for closing remarks. Ma'am.
So am I audible? There is some network connection issue.
Yes, ma'am, you are audible.
So as we come to the close of this insightful webinar, I would like to extend our heartfelt thanks to all the speakers for their valuable contributions. Your insights have shed light on the immense potential of this subsector of biomanufacturing for mRNA therapeutics. Finally, please mark your calendars for the next webinar in this series, which will continue to explore various thematic areas of biofoundry and biomanufacturing initiatives. Details will be shared shortly on the DBT and BioRack websites and through various social media handles. Thank you once again to all the speakers, participants, and the organizing team for making this event a success. We look forward to your continued engagement in the upcoming sessions. Thank you so much.
Thank you.
Thank you.
Thank you.
Thank you.